Good afternoon, everybody. This is Nicholas from ISP Singapore here. Welcome to the first Technical Tuesday of 2022. Some of you may be wondering why am I here, because I'm temporarily taking over from Linda. She's currently busy at the moment, so I'm just standing for her as part of the EXCO committee members. Yeah. So thank you all for joining us for today. Today we have a special session, and the topic is actually control of cephalosporins cross-contamination in pharmaceutical manufacturing, and that's a risk-based approach, right? So for today's session, we have our guest speaker. It's from Pakistan, and his name is Aqeel Ahmed Sumro. He is a doctor in pharmacy and as well as an MBA. He's the expertise in all pharmaceutical dosage forms. I'll leave you to listen to him on his detailed profile later on. But the main thing is that we are here today to learn from him. So at the end of the session, there'll be a Q&A. So feel free to post any questions that you may have during that time, and we will address them accordingly, right? Yeah. So without further ado, I will pass the session over to Aqeel. Aqeel, over to you, please.
Thank you, Nicholas and Christina. I welcome all, and good morning, good evening, and good noon to everyone. Today we are here to have some session on risk-based approach for the control of cephalosporin cross-contaminations in a pharmaceutical manufacturing sites. As far as my introduction is concerned, I'm Aqeel Ahmed. I belong to a pharmaceutical manufacturing industry, approximate 20 years work experience in pharmaceuticals. Currently, I'm associated with Brookes Pharma, located in Karachi, Pakistan. And my area of expertise are sterile manufacturings, and especially sterile solids and sterile liquid manufacturings. And also I remain the part of beta-lactam sterile manufacturing facilities. So we are here to discuss something about the cross-contamination of cephalosporins. The content today we will discuss, first of all, we will go through the beta-lactams, or in the beta-lactams, what are the cephalosporins. In detail, we will learn what are the cephalosporins, then the cross-contamination and the risk management. There are three basic aspects are there: the risk assessment, their controls, and in the last, the risk review. In the risk assessment session, we will understand aspects to be reviewed during risk assessment, the description of the cephalosporin facility, and the description of the risk. And for the controls, we apply over there for the engineering and procedural controls, their process flows, personnel flows, document flows, and comprehensive monitoring program. After that, we will review the risk. It's a continuous process, and in the last, we will conclude the session. So let's start. This picture is a worth of 1,000 words. Definitely, we are talking about the contamination controls, and especially for the cephalosporin contaminations. Then the answer goes for the patient safety. The main objective to conduct this session and give the risk assessment basis is to save the humanity, the patient safety. By controlling beta-lactam contamination, we can save the humanity. Now, the question arises, how a person can save the humanity by contamination control saving. So let's start. We all are well aware about these diagrams. So approximate 10% of population is hypersensitive to the beta-lactams. Now, what are the beta-lactams? Basically, beta-lactams are bicyclic rings are there, just like this. Bicyclic ring is there, and it is consists of so many components. For example, just penicillin. Penicillin is the one of the component of the beta-lactams, then the cephalosporins, penems, carbapenems, and manobactams. In these ampicillin, oxacillin, cefixime, cefepime, cephalosporin, fluorocarb, penams, all belongs to the beta-lactam categories. Now only we will discuss to the cephalosporins. So basically, if we go for the beta-lactams, they are one of the largest group of the antibacterial products used clinically to combat the most resistant pathogens. All beta-lactams have a characteristic bicyclic core structure, which is believed to initiate allergic reactions. So whenever you see this type of the structure, you should understand that there is some allergic reaction over there. Any component which contains this type of the structure, which would lead to the hypersensitivity or it will lead to the allergic reaction. So cephalosporins are among the most prescribed antibiotic classes due to their wide clinical utility and general tolerability, with approximate 1 to 3% of the population reporting a cephalosporin allergy. Overall, beta-lactams have 10% due to the penicillin. Penicillin is the most potent component of the beta-lactams, which produce approximate 10% of the population is hypersensitive to the beta-lactams. But we go for the non-penicillin beta-lactams, there is a range. In that, there is the cephalosporins, penams, and others. In that, cephalosporins contain 1 to 3% of the allergic reactions to the whole population. It depends upon either it will go for the urticaria, it go for the asthma, or it may go for the some other reactions, any rashes on the body. So it depends upon the demographic conditions. Either we go for Pakistan, it may produce some urticarial reactions, but it will go for the West or in the America side, they may have some another type of allergic responses. Maybe there is the asthmatic allergic. So it depends upon the area. It's not necessarily that same type of symptoms will be produced by the cephalosporins. Yeah, this is the same ring, beta-lactam ring, and we are here to see this thing. So now it's about the beta-lactams. What are the beta-lactams? We now clearly understand what are the beta-lactams. And in the beta-lactam, there are 4 types of the things. Penams are there, cephalosporins are there, and penicillins are there. So penicillin is the most sensitive. So other than penicillin beta-lactams, there are cephalosporins. So today we will discuss only one component. It's cephalosporin. And cephalosporin widely available in so many dosage forms. It's available in tablet form. It's available in dry powder injectables form. It's also available in capsule form, and it's available in the dry powder injectables. So today, if we go in the depth and we will discuss a case study, it will majorly pertain to the injectables. So before we start the cross-contamination risk assessment, so just go through the cross-contamination. As per definition of the cross-contamination is the contamination of starting material, intermediate or finished products with another starting material or the product. It means if we go for the starting material, it means the raw materials, intermediate materials, semi-finished, and in the finished form. These 3 types of the materials can produce contamination to the another product. It means cephalosporin contamination with another non-cephalosporin products contamination. So these 3 stages of the material can do the cross-contamination. So far, manufacturer must have a process in place to not only avoid contamination scenarios, but also provide documented evidence that contamination has not occurred. So we have to plan a documented risk assessment, and we have to put some controls over there, and we have to review the risk. Where are the processes where the contamination can be started. This is ICH-Q9 quality risk management process. This is the diagram, a life cycle of the quality risk assessment. So if we go for the definition of the risk management, it's a systematic process for the assessment, their controls, their communication, and review of the risk. This definition given by the ICH-Q9, a risk management program starts with identifying the possible risk associated with the product or with the process used to develop, manufacture, and distribute the product. So as far this life cycle diagram is concerned, it's distributed into 3 stages. Basically, risk assess. We have to assess the risk. What is the risk? As far as in this case, this is the cross-contamination, and it may go for the asthmatic reactions, or it will go for the allergic reaction. This is the risk. It may go for the it will even go for the life-threatening. So we have to identify the risk, the source of risk. Then we have to analyze it. What is the potential risk? What are their acceptable limits? And what potential risk we can bear? Then we have to evaluate it. Then we have to put some controls to mitigate the risk. So to reduce the risk and their acceptable limits to be there. And the both strategies we have to communicate cross over the departments and with all the engineering from quality control, quality assurance, production, engineering departments. We have to keep it communicated over there, and we have to document it. Then in the last, we have to review the risk. Periodically, we have to review the risk. What are the review? There should be some key performance indicators which indicate us for their limits, for their actions. So further in detail, we will discuss this. As far as the risk assessment and their identification is concerned, first of all, we have to identify the risk. We have to analyze it and then evaluate it. As all products containing hazardous substances are not equally potent. It means in the cephalosporin, even all are not equally potent. So we have to describe the limits, acceptable limits. So risk assessment to be carried out, determine the potential hazards to operator, to environment, and their cross-contaminations. So there are basically 3 types of risk over there. It may go for the operator health, it may go to the environment, and it may go for the cross-contamination in between the products. So let's check out what worst case could be there. What are the types of risk over there? If you go in this case in pharmaceutical cross-contamination due to the cephalosporin, death case may be there. Permanent minor injury, consent decay, recalls of the products may be there. In case any ooze and oot are there, but sometimes companies has to discontinue their products. Litigations cases are there, product integrity loss, potency loss may be there, complaint, out of trend results, reversible major health issues, reversible minor health issues, observation during inspections. Some inspector regulators may put some observations, and you have to put their COPPAs and other things. Out of specification results are there, permanent major health issues are there. Sometimes it goes for the even business closures, warning letters from regulators, cessation of the business, and regulatory non-compliances for the sale. So these all are the risk associated. So we have to analyze that due to this issue, which type of the risk we may carry. So in this case, we have to analyze. It depends upon the facility. It depends upon the dosage form. It depends upon the control you applied. So these are the risks. So let's go again. So in the assessment, you have to plan how you have to calculate your risk. So most potent and hazardous product or API to be selected, as in the cephalosporin, you may select the ceftriaxone sodium, or you may select the cefotaxime sodium. So it depends upon, you may go for the cefixime. It depends upon either you are manufacturing for the tablet, you may go for the cefixime, or you go for the dry powder injector suspensions, cefaclor are there, and there's some materials are, it depends upon the facility. So which type of the API you are using, and what is their potent risk behind there. So I selected for this case study, it was the ceftriaxone sodium. It's the dry powder injectables over there. So then after, you have to selection of risk assessment tools. It depends upon the well-defined problem description or risk questions. Types of information needed to address, and their acceptance limits and actual data of cephalosporin contamination to be established. And what would be the analysis method? It should be declared before analyzing the system. It may go with the swab testing. It may go for the environmental samples are there. Some sensors are there. So it depends upon. So you have to declare what tool you will use and what analytical method you will choose to analyze your risk. Definitely, we have to go for. We will understand the risk and we will take some samples and we will check out their concentrations and we will decide either it is allowable or not allowable. As far as the tool is concerned, so basic risk management facilitation methods, tools are so many tools are there. The flow charts are there, check sheets, failure mode, effect and criticality analysis, failure mode, effect analysis, and free analysis. Sometimes risk ranking and filtering tool you may use. So it depends upon which type of the risk you are you would like to calculate and what data you have, what type of facility you have. If we go through the Fema, this is the Fema form, and this tool I use for that. I just gave you some example for only understanding it. It's not a benchmark. It depends upon facility and it depends upon the risk you are going to assess. If you go for the process steps, it means here is the risk of cross-contamination. If you go for the potential failure mode, it means in what way could be step change to feature go wrong. For example, here we are, the main could be the major source of the cross contamination. So you have to put some controls over there. The potential failure effect, what could be there? But because if a person is here, which is the source of cross contamination, it means the source of contamination of cephalosporin, and we all are known that cephalosporin is a potent component and 1 to 3% of the population hypersensitive to this. So this is the risk. So now you have to put some potential controls over there. Definitely a person is going for from cephalosporin to non-cephalosporin, then should be a gowning procedure over there. Decontamination procedures to be established over there. So it depends upon you which type of the tool you are going to use. It depends upon your sort of their potency, raw material potency, and their daily intake. This is the one of the example for the manpower. It can be the leakage in the HVAC system. In case if there is any leakage in the HVAC system in the duct, it may go contamination may go to the environment directly, and in the other end another facility is there and it may suck from the suction of the another facilities inlets, so it may go. So you have to put some controls for the analysis of even ducts also. Their periodic reviews should be there and something else to be there. When we assess the risk aspects to be reviewed during risk assessment, so definitely their design and construction of the structure we have to check. Their chemical and physical containments controls you already applied. We have to review these all also. Process operations, their acceptance norms and assessments, and material and process flows to be checked out over there. Yeah. Here is some understanding the evaluation of the risk. Evaluation of the risk to quality should be based on a scientific knowledge and be ultimately linked to the protection of the patient. This is the thing. So what is the your accepted level? Which component you are using over there, which type of API you are using over there, what is their acceptable intake, daily acceptable intake level, and what is their cross-contamination levels. So you have some scientific calculation over there. The level of effort formally and documentations of the QRM process should be balanced with the level of risk. Normally, potential risk is relation to the following should be considered. It may be the material and due to the ingredients, physical characteristic and composition of the product, the process procedures, microbial limits, where applicable equipment, premises, packing materials, sanitization and hygiene, personal human errors, and utilities. So the selection of raw material to whom you will assess the risk. For example, in some capsules facility, there is a cephexime, so it would be cephexime, the calculation or the risk assessment or the swab testing analysis would be for the cephexime. If some facility would like to work for the dry powder suspensions, so it may work for the cephalosporin, cephexime also, or cephaclor also, or another APIs may be there. So these are potent APIs and moderate synthesizer agents and high potent substances. Occupational exposure limits, OEL, for the 8-hour shift, a person's working over there, their acceptable daily intake ADI and swab limits should be set as a moderate synthesizer agent. So moderate synthesizers means skin irritations are there, their allergic response may be there, and a high potent substance into the category 3. So the raw material selection depends upon your facility, which type of study you would like to conduct over there, and what is their acceptable limits. This is the scenario when we will discuss some facilities are fully dedicated and there isn't any no relation in between two facilities. So these are the independent departments, so independent facilities. There isn't any risk of cross-contamination. But I'm talking about those facilities which are on a same site, having a same different buildings, but there is some shared areas over there. So although cephalosporin and non-cephalosporin facilities are dedicated and located in two separate buildings, but some of the operations of both facilities may done in a shared or on a common area. For example, in a cafeteria, persons may meet, and quality control departments, quality assurance, engineering services, warehouses are there. It depends upon the facility where you are working. Maybe in some facilities you have a separate cafeteria for cephalosporins and non-cephalosporin, and quality control lab is there. But there are some facilities there. This is the common areas over there. Separating the non-cephalosporin facility from the cephalosporin dry powder injectables facility, a risk-based approach is required to avoid cross-contamination in accordance with GMP. So here we will go through some layout of a facility. So a pharmaceutical manufacturing site having cephalosporin and non-cephalosporin facilities, then what kind of risk-based approach or measures and controls are required to prevent cross-contamination in between both facilities? Now in the yellow color, this is the cephalosporin facility. In the blue, it's non-cephalo, and there are some shared areas. This may be that some park areas are there, and this may be the cafeteria. So there are the chances of cross-contamination in between this facility and in between this facility. There are chances of the traces of this facility may come into this facility, and it may cross-contaminate the journal products. So there should be some controls over there to avoid the cross-contamination in between both facilities. The distance, their layouts, their procedural controls, their document flow controls, their engineering containment is there, their HVAC system, the inlet and outlet face-to-face of lamina and HVAC systems. So many controls are over there. It depends upon your facilities and it depends upon your product, what product you are going to produce over there. So if we discuss on a case study for injectables of dry powders, so it depends if it's a description of a facility. If I discuss what are their controls over there. So there is a dedicated facility for the cephalosporin dry powder injectables only. There is a separate building. The HVAC is separate. Main material flow and there is control. There all are separate. They are using pre-sterilized cephalosporin dry powders of 5 kg or in a 10 kg of containers. They use containments. Their plant is very highly sophisticated and very good containments are there. Split butterfly walls for connecting sterilized powder, filled containers, proper interlocking system is there. Building management system is working over there. Safe bagging system in HVAC. Appropriate air pressure and alarm systems are there. and terminally mounted HEPA filters are there. So this is just a description of a facility, which are definitely a controlling tools also there. So here we will see the description of the risk and controls. Following may be outcome because cross-contamination between cephalosporin and non-cephalosporin, basically main material, mother nature, and machineries. Basically, the movement of these all the things is the basic source of cross-contamination. So men can move in both facilities and in a shared area. So this is the main critical aspect for the transportation of the cross contamination. If we control this person, there isn't a dedicated persons over there. Their dedicated uniform color coding is there. Their route of in and out is separate. So we may control this. Then there is the movement of the material in both facilities. Some pallet may be used, some buckets may be used, some lifting equipment, just like a lifter and trucks are used. So their layout, their in and out routes, and their distance, these are the areas where we can control. So we have to check these all the things. And the movement of the machinery, for example, if there is any machinery, for example, you have the lifter over there, or after few there isn't any use of that machinery in cephalosporin. Now, how to transport this machinery into the non-cephalosporin building? So, what would be the mechanism for their decontamination? These all the procedures should be established over there. And the environments of the both facilities, it means that the suction of if you go for the big slide, the suction of this should be not face to face to this facility discharge. So we have to check this thing. So procedural controls are required to eliminate the cross-contamination of cephalosporin and non-cephalosporin by keeping in view the personnel flow, material flow, document flow, and their operational rules are there. So as far as the personal flows are there, this is one of the control. Person who leave the cephalosporin area should change clothes, their shoes, and wash their hands and other things. A person who enter in cephalosporin area cannot enter a non-cephalosporin facility on same day. In case of emergency, additional controls are required to take a bath or showers or change their clothes and take some time, and something else controls to be provided over there. These all are the in-house controls over there. And the third, if an individual requires to enter in both facilities on the same day, so he should first enter into the non-cephalosporin facility, then to the cephalosporin facility to reduce the cross-contamination. Availability of bath or shower facility should be there to reduce all the things. Yeah, this is the graphic presentation for the control of personal flows. Here is the cephalosporin facility. This is the shared facility, and this is the non-cephalosporin facility. This is the shared facility. A person can go to change their clothes and wash their hands and shoes, and he can enter into the cephalosporin facility. But this person cannot go to the non-cephalosporin facility. This is one of the personal control over there. A person working in non-cephalosporin facility, he can come in this facility. He can he may go to the cephalosporin facility also, but he have to change their clothes, their shoes, wash their hands, and some controls should be there. But this person cannot back to this facility on the same day. A person working in cephalosporin facility, because it's a potent component over there. So a system for government decontamination from leaving the facility to be provided. How to decontaminate your garments, their washing procedures should be there, and their soiled uniforms putting areas should be there, and their proper disposition to be there. There should be a facility for the change of cloth, air shower system to be there at the in and out of the facility. Somewhere some mist showers are required, or water showers are even required if a person go to another facility. And some appropriate devices are also required, just like door interlocking system is there, their building management system is there, their security systems are there. So appropriate devices are also recommended for the decontamination of the system. Then after human or personal flow, there is another thing which may be a part of the cross-contamination. It may be the source of the cross-contamination. It's a document. A document written in cephalosporin facility should not be brought into the non-cephalosporin facilities. The document written in the cephalosporin facility should be sealed up in a clean, dedicated, color-coded bag with when it is brought out from the facility. The document sealed up should be opened only in a dedicated room for the review and storage. If we go for this graphic understanding, this is a cephalosporin facility and this is the non-cephalosporin facility. The document can be write and read and seal in a pouch. It's a color-coded pouch, and it can only and only transfer to this shared area. Their storage place would be there, their reading and review place would be there, sealed envelope in a pouch, case, or a read-only documents are there. But this document cannot transfer to the non-cephalosporin facility. But because this document contains some traces of cephalosporins, we put one control that we put in a clean bag and it's a color-coded bag. Then this document only can come out to this shared area. After that, there are chances of some traces over there. This document cannot go over there. But because here if come, it may leave some traces of cephalosporin, which may become the part of non-cephalosporin products. The document which are written or read over here, they are only transferred over here, and these are also cannot go to this area also. So this is the control for the document or some other type of materials. And same go for the packing materials or something else. After that, one thing is that the material flow and their operational controls to be there. So material or product brought out the cephalosporin facility should be enclosed in a dedicated container and it should be sealed up. Dedicated pellets to be provided for that. There should be buckets duly color coded to be used over there. So these both maybe become the source of cross contamination in between both facilities. So no material or equipment to be transported from cephalosporin to non-cephalosporin facility. Launder could be done outside with third party with agreement to wash in a separate dedicated area and their equipment. You have to evaluate your launder and you have to visit it, you have to approve it, and you have to check out that either he's not using the laundry washing of another company while they are using these potent components. So you have to keep check on there. You have to periodic review over there. You have to visit the launder and you have to approve the vendor. So third party, if you go for the third party, if you cannot bear the third party expenses, you have to put your in-house laundering system. So washing of your laundry, either it will go for the sterile laundry or if it's a journal laundry, now you have to make some arrangement for the in-house washing. So it is also a source of cross contamination. The tools and measurement instruments used in the maintenance or in a calibration to be dedicated and duly coded. So it's also a chances of when a vendor come for the calibration of the equipment, they go for first into the cephalosporin facility, then they go for the non-cephalo. It's wrong. So you have to put some procedural control over there. First, you have to go with the non-cephalosporin facility. Then you have to decontaminate your equipment. Then the vendor, third-party vendor, can put their instrument to the non-cephalosporin facilities. Waste material to be separated and sealed inside the facility and proper disposition to be there. Separate in-house laundry for cephalosporin contamination. Uniforms are there. The effluent treatment should be there. So these are material flow and their operational rules through which we can control the cross-contamination. After that, when we put our containments, we put our controls over there. Now we have to monitor the contamination. Now, how we will monitor our cross contamination? Why we are doing this thing? To monitor and risk control, there must be a management of a high and significant risk, along with a contingency and fallback plan if it is required. Team should ensure the risk management plan do not create additional or residual risk, and future outcomes should be assessed and followed up on with breakthrough risk examined to determine how the process can be improved. Dynametrics should be reviewed that provide insight on performance, their trend, and potential risk. Stakeholders should be communicated with on risk management efforts. You have to review these all statistics, and you have to put some KPIs over there. You have to put some key indicators which give you the feedback of what is the level of cross contamination going on over there. So when you plan your comprehensive monitoring program, so a comprehensive monitoring program is required to evaluate the compliance and appropriateness of the control. Scope of monitoring should be on both facilities. Monitoring program to be took in place, including periodic sampling, product testing, and surface sampling. If you use swab sampling as a tool for monitoring of contamination, it's a very good one. So you have to elaborate their procedures. The product contact surfaces area which are most difficult to clean to be selected as a sampling point. The location selected for swabbing should be generally those locations that are most difficult to clean. Very confined areas you have to select over there. The representatives of different materials and representatives of the different functional locations, the side corners and handles of doors, and so confined areas are so many there. If these locations are swept and if residues in these materials are acceptable, then residues on another clean location would be also acceptable. Floor with high traffic zone at cephalosporin facility and exhaust outlets and other facility exhaust inlets. You have to take swab from exhaust inlets also and the exhaust outlets of the cephalosporins also. There should be some limits for acceptable limits and their action limits. So staff, if you go for the sampling, swab sampling, you have, I recommend, it's up to your facility. It depends upon you. Some GMP entrances, floors, walls, handles of doors, the facility corridor floor, and footprint. If you take some footprint steps away from the staff change room towards the general facility or shared area, you have to take some swabs from 50 steps away from the facility, 100 steps away from the facility, 150 and 200 steps away from. It depends upon you. If there is a high concentration, you may go beyond that. When you go for the 300, 400 and 500 steps away from that. So you have to put controls to reduce this. Dust collectors of the cephalosporin facility, inside that dust collectors, what is the concentration over there? Exhaust ducts inside the cephalosporin facility, and finished goods exits. When finished goods go out, it also carries some traces of cross contamination. Maybe the source of finished good, maybe the source of cross contamination. So you have also checked this swab sampling at the 50 steps or 100 steps away from the finished goods stores or from the finished goods where it exits from the facility. You have to plan some swab sampling from the cafeteria, which is also a shared area, a non-cephalosporin facility, just as HVAC inlets. After putting these controls and sampling plans and their sampling and understanding of that results, and then you have to review the risk. Periodically, you have to review the risk. So when you assess the risk, you put the controls over there to you mitigate the risk. Now you have to periodically review the risk. A long-term scheduled monitoring program is required in both facilities. The monitoring program to be planned to take swab samples, monitoring schedules to be predefined as it the month of first month, second month, third, sixth, and after each year, 12 months, you have to onward check out the sampling or you have to review the data. In addition, in case of unscheduled events such as the remolding of the facility or removing of any equipment, the monitoring program may be done accordingly. This approach to managing contamination control, it includes the creating targeted and risk-based measurements of contamination avoidance, implementing key performance indicator to assess status of contamination control, and ensuring a defined strategy for deviation management and investigation, including corrective and preventive actions and their change managements. So you have to keep reviewing your risk. You have to monitor your risk by reviewing and they are monitoring. You may be on a track to evaluate your risk, the whether either it is within limit or it's out of limit, or either we lose our controls or we need some additional controls. It depends. It's a continuous ongoing process. Here are some key performance indicators which should be kept in review. Or, for example, deviations of the area, you have to keep a control in there. It may give you indicator that there is some contamination cases of any deviations due to some contamination cases. You have to review it. Some ooze results of the products in non-cephalosporins, any product recalls or any OOT results are there, out of trend results are there, any results in terms of swab sampling and OOT results are there, then you have to be focused for the again rechecking of the system, any leakages in the system, any deviation in the system, any market complaints or any market allergic complaint from the pharmacovigilance point of view. So you have to keep some KPIs for your review. So it depends upon, it's not only recommended by me, it depends upon which sort of the containments you would like to be there and which KPIs you are going to be checking, you are keeping for yourself. So in the last, we will conclude our session. Risk is unavoidable with the number of moving parts in the facility or a laboratory. QRM program can help minimize the risk and maintain the acceptable level of the risk. Reviewing, identifying, monitoring, and developing responses to risk or threats, an important step toward the QRM implementation. Each department has its own role towards the risk management. Either it is an engineering department, it's a quality control department, quality assurance department, a cephalosporin department, and a non-cephalosporin department. When their facilitation systems are there, it depends. So it is the role of whole the company. Through taking these steps, one could establish a qualified control system to control cephalosporin cross-contamination in pharmaceutical manufacturing. This was a general strategy for the controlling the system. They are establishing the QRM, the documentation of the QRM, their reviewing of the risk. I recommend if you have this sort of the controlling over there or this sort of the management, you can put this same controls to other potent components, just like hormones or other items are there, psychotropics are there, or something else are there. So this is generally a basic review for the risk and cross contaminations. So Nicholas, it's about to end now. So it's question answers are welcome over here.
Yeah. Okay. So thank you very much for the insightful presentation, Aqeel. Right now it's the Q&A segment. So right now there's basically two things, right? So feel free to actually pose your question directly to Aqeel right now. Unmute yourself and introduce yourself, and ask him directly the questions. Or the other alternative is actually post your questions in the chat. I think right now we have a few questions, so I can come to that first. And secondly, if let's say if you have no other questions, or you finish asking the questions or have no other questions, you can do the second thing, which is a survey, right? So we would like to get your inputs on this session so far, and how we can actually improve for further sessions. Yeah. So it's just basically one or two of the things that you can do right now. So without further ado, let me go on to the first question. Aqeel, I need you to address right now. So that's been that we have received in the chat. Question number one: how often are the facilities monitored?
Sir, this is an ongoing process. It depends upon the infrastructure of your facilities and the selection of the component you are producing over there. Their dosage forms, it depends upon you. It is the validation process, their cleaning validation process is there. Usually, I already told in my slides that initially you have to check these, so you have to take swabs after 1 month, after 2 months, after 3 months, and after 6 months. And ongoing, after each year, you have to monitor the facilities.
Okay, thank you. The second question is, could you explain, well, more in detail about the footprint and 10-50 steps on the floor? I'm just reading out the question.
It is general. Yeah, it's a good question over here. First of all, we have to. This is our in-house procedure. It depends upon your facility. Either you take the samples from 50 instead of 50, you take it from 100. It depends upon you. It's depend upon your facility and your controlling systems. So the swab sampling at the footprints, it depends upon your facility, and you have to establish something over there. You have to write some steps over there. If you get satisfactory result at 50 steps, it's okay for you. But if in spite of 50 steps, there is a contamination in 100 steps, then you have to put some controls over there. It means your staff is not changing their shoes, or the shoes are being same used in the both facilities. It depends upon you. So how you are using over there may be some in and out, more than in and out. There isn't any containment air shower system over there or something else over there. So it depends upon your facility. You have to in-house generate these limits, and you have to generate these steps distance over there. Moreover, as far as the 10 ppm is general cleaning requirement. So general cleaning, if it is under 10 ppm, it's allowable. So it's a minimum contamination which is allowable. It's a 10 ppm. It's a general cleaning validation rule for all the potent materials.
Okay, thank you. Next question. We have a few other questions. We've got a list. Is it a requirement to have separate water systems? What are the risks that need to be considered?
Yeah, I suggest for separate system, why? Because separate loop systems are there, but sometimes in the loop there is a negative or return pressures are there. So then there is the chances of cross-contamination. The journal and the mix-up of the cephalosporin traces into the journal flow of the water. So it is in-house, but it's a recommended. It's not guided in any guidelines, but it is journal recommended by experts that it should be separate.
Okay, thank you. Next question. When assessing raw materials, what do we decide and what is the next step?
Sorry, please repeat your question.
Okay. When assessing raw materials, what do we decide and what is the next step?
If raw material, it goes to the warehouse, definitely where pre-cleaning is required over there, and dedicated warehouses should be there. The raw material of cephalosporins to be stored in a separate warehouse and to be used in the same facility. But as far as the return packing materials are there, it should be also used inside the facility and or disposed of. Sorry.
Okay. We have 3 other questions here. Next one. Would you please tell us the reference of the swab limit, not more than 10 ppm?
Definitely, sir. It's an in-house procedure. There isn't any expert's opinion is there. Experts suggest that you monitor your facility via tracing the swab samples of the floor or the walls or the handles of the doors, and you have to check on a spectrophotometer or HPLC, and then check their concentration. You may make the reference with WFI or water. So it's depends upon you what how much concentration of cephalosporins are there. So basically, it's a general in-house procedures.
Okay. Next question. Are there any risks to chemical tests if cephalosporin and non-cephalosporin are tested as shared facility?
I cannot. But the analysis of the raw material, this is the work in the quality control department. And quality control department, if it is used in a shared facility, it's contaminated with cephalosporin. So it is recommended to make it separate, and it facilitate to put some 1 or 2 HPLCs in the cephalosporin building, and their testing to be carried out over there, and their equipment to be separated. It's recommended. But cephalosporin, if it is a shared facility, then it is the contaminated area with cephalosporins. So any movement of the material from quality control department to non-cephalosporin building, it may contaminate to the non-cephalosporin building also.
Okay, we have some more new questions coming in through the chat.
Yeah, welcome.
Okay, yeah, I think it's very interesting for most people here. Next question: can we use? I think he used decontaminated solution. I think it's cleaning solution.
Yeah,
can we use decontaminating? Sorry, just hang on here. Can we use decontaminating or cleaning solution? Example, sodium hydroxide 5% for cleaning tools or facilities before moving from cephalosporin area to non-cephalosporin area.
Yeah, it's true. It's recommended to use sodium hydroxide. It depends. Recommended is 10%. So you can use sodium hydroxide 10% solution for the decontamination of the equipments or decontamination of the articles being to be transferred from cephalosporin to non-cephalosporin building. And then after decontamination, you have to take the swab samples and you have to check out their concentration. If it is less than 10 ppm, then you can allow this equipment to be shifted into the non-cephalosporin facility.
Okay, next question. What about the storage of finished goods? Should it be separate, or is it in the same together, or is it the same when in separate area?
Sir, finished goods stores, it depends upon, but because there isn't any chances of contamination over the cephalosporin finished products, you may store both in the same. But there should be no transportation in between warehouse, finished goods warehouse, and non-cephalosporin buildings. In the finished goods, there is two minimum chances are there for cross contamination, but because if you put the air shower system at the exit of your finished good, then the traces of which are available on the surface of the container, it will be removed, or any vacuum suction system to be there. So the finished outer surface of the boxes to be cleared. So you may put it into the warehouse also.
Okay, the next question is more of a statement, but I will rephrase it to a question instead. What are the risks for chemical tests?
Risk of chemical test?
Yeah.
I can understand we should separate for micro test, but I'm not sure to risk for chem test. This one, you are?
Yeah, or maybe the speaker himself,
Okay.
the person asking the question can ask.
A question I'm seeing over here on the chat box. It's I can understand we should separate for the micro test, but I'm not sure of risk of chemical testing. It's depend. Chemical testing means the persons coming from non-cephalosporin to quality control lab, and person coming from cephalosporin building, it also comes to the quality control lab. So quality control lab is totally contaminated with cephalosporin. So it may become the source of contamination to contaminate the non-cephalosporin building. So either we perform the chemical test over there, or we go for the macrobiological test over there. For microbiological testing, there are now available disposable petri dishes are there. So that maybe we may mitigate the risk of cross-contamination for the microbiological testing equipment also. So there's build in a shared facility where the quality control department is the same, the quality control lab is contaminated. It may be the source of cross-contamination.
Hi. So it was my question. I just want to ask, I can understand for microlab, maybe if there's contamination, it might mask the growth of bacteria, right? And maybe we get a false low count or something. But what if the setup is where the chemistry lab is inside the cephalosporin building and it tests for cephalosporin and non-cephalosporin samples? Is there any risk to the chemistry lab chemistry test?
My answer is same, which I have told already. Putting some setup in cephalosporin building for that chemical analysis, it will be more better and it would be more controllable step. Because when we test, either you go for the chemical test or you go for the microbiological test in a same quality control lab, which is shared in a shared facility. So a person coming from non-cephalosporin may be contaminated, or the equipment which you are using in non-cephalosporin, the samples coming and out, the person's movement from the quality lab to non-cephalosporin building, it may carry on the traces of the cephalosporins. So the quality control lab, whether it is used as a shared, it may be the source of cross-contamination.
Okay, so that's from the perspective of material and personnel flow. But for the chemistry test itself, there is no risk, right?
There isn't any risk. I don't think so.
Okay. Okay.
Thank you.
Yeah, thank you.
Okay, thank you. We have one more question here. Can cephalosporin and other bacter-lactam be manufactured in the same facility?
As far as non-penicillin items are there, it is recommended to manufacture them separately, because cephalosporins and penicillins cannot be manufactured in the same site. Even penims are not recommended to be manufactured in the same facility. So the cross-contamination in between cephalosporin chances are there, or cross-contamination in between penicillins is there, but because penicillin is more potent, approximately 10% of the population is hypersensitive to the penicillin, and with the cephalosporin it's only 3%. So penicillin can be the part of it's a more potent, and it can be the more chances of hypersensitivity to cephalosporin contamination. So it is advisable to make the separate facilities.
Okay, I think we have someone who just raised a hand to ask a question. Yeah, you can go ahead to unmute yourself and ask.
Thank you. This is Ghazan for Javed. And my question is that thank you for a very insightful presentation, and Aqeel and in ISP for organizing it. I had a query that do you think the cephalosporins can be manufactured with the oncology sections or not? I'm not sure if this question was asked earlier or not. I mean, in the same, let's say in the same facility, but with some distances.
In the same site, it can be manufactured, but the facility should be separate. Their
and how much
should be there, and their in and out layouts should be separate.
Okay.
It's just my expert opinion.
Yeah.
I will back you with this question.
Thank you.
Thank you.
Okay, I think probably we can go on to our last question here, since it's already 6:30. Now, is there another sampling method?
It could be some sensor may be there, some electronic systems may be there for the checking of the traces of the cephalosporins, but recommended method is for qualitative and quantitative analysis is swab sampling.
Okay, so thank you very much for addressing those questions. I think due to time limit, we will have to end here, unfortunately. But feel free to email your questions directly to Aqeel, and I'm sure he can address them separately. Yeah. So just two last things to end off. One is that just a reminder, we'd like to get your inputs for the survey that Christina has actually posted in the chat earlier on. So this is actually to help us improve for this survey, for this session, as well as for future ISP webinar or sessions like this, right? And yeah, Christina has just reposted that. That's that. And of course, the last thing is that now we are talking about cross-contamination in this webinar. So stay tuned for future webinars from us, especially when we talk about cleaning validation. Yeah, so just stay tuned for that. So that will be in link with what ISP is presenting down the road. Yeah. Okay, so yes, so thank you very much again, Aqeel, as well as all the other attendees who have posted your questions. Very nice and insightful. So we hope to look forward to see you again for future webinars. Yep.
I also thank on the behalf of my team to all participants and ISP Singapore team, especially to get Nicholas, Linda, and Catherine. So thank you very much for that. Yes, someone.
Thank you, Aqeel. Thank you.
Thank you. Thank you very much.
Thank you.
Thank you very much, Nicholas and Aqeel.
Thank you.
Thank you.
Bye.
You can contact me at my this email address, or my WhatsApp number is there. You may have questions over there.
Yes,
Thank
and we
you,
will also
Linda.
share. We will share the details as well in a follow-up email. With
Thank you,
everyone.
Christina. Thank you,
Thank
Christina.
you. Bye.
Bye.